Lastly, you should be familiar with three types of polycyclic ring systems that you may encounter in your textbook. These are the spirocyclic fused ring and bridged ring polycyclic ring systems. Basically, any polycyclic ring system is a ring system that includes more than one ring of various sizes. Although I'll include the IUPAC naming system that's used to name each of the polycyclic ring systems you've seen here, you're not expected to know that for this class. You should be able to recognize the different types of polycyclic ring systems, though, when you see them. So first off, a spirocyclic ring system is one in which two separate rings are connected together by a shared atom. These are named as spiroalkanes, and the numbers in the name indicate the numbers of carbon atoms that are unique to each ring. You can see here in the structure that I've drawn, spiro-4,5-decane, a six-member cyclohexane ring shares a carbon in common with a five-membered cyclopentane ring that's circled on your slide. Bridged ring systems, in contrast, share non-adjacent atoms that are common to two rings. What this does is it creates a sort of a structural bridge, as you can see here, 1, 2, on top of this cyclohexane ring on the bottom. This is a bicyclic system that contains only two rings. You can tell that by cutting the ring. The minimum number of cuts you need in order to produce an acyclic system tells you how many rings there are. So if you can cut the bond here between 1 and 2, you'll get rid of one ring, leaving behind a dimethyl cyclohexane. If you cut any of the bonds in the cyclohexane ring at the bottom, you would get an acyclic system. That's how we know there are two rings here. Bridged ring systems are named as bicycloalkanes. Here I have bicyclo-3,2,1-octane. The numbers in this case, in decreasing order, indicate the number of individual carbon atoms that is in each bridgehead. So 3 carbons in this bridgehead, 2 carbons in this bridgehead, and 1 carbon in this bridgehead down here. There on the slide are indicated the 2 carbon atoms that are shared between the 2 rings that create the bridged system. Lastly, we have fused ring polycyclic systems. In fused rings, adjacent atoms are common to 2 rings as well as a shared side. So on this slide, I've circled the shared side that's common to both rings. These are named as bicycloalkanes. Here I have trans-bicyclo-4,3,0-nonane. In fused ring systems, the third and final number in brackets is always 0 because there's no carbon atoms in the shared side between the 2 rings. Notice that fused rings can also have stereoisomerism. The indicated hydrogens on this slide are both on wedges, which means they're on the same side of both rings. Alternatively, we could have a stereoisomer where one of the hydrogens is on the opposite side from the other hydrogen. So I just want to end this pre-lecture by saying, who cares? Why should I bother learning about polycyclic carbons? Why should I recognize fused ring systems when I have to worry about conformational analysis and naming different types of cycloalkanes? Well, I want to point out that many biologically important molecules are based on polycyclic carbon skeletons. So for example, human steroids are often based on this carbon skeleton structure that you see here, which includes a polycyclic fused ring system. 3 cyclohexane rings fused together, and a fourth and final ring over there is the cyclopentane ring fused to one of the 3 cyclohexanes. This basic structure is found in human sex hormones such as testosterone and estradiol, which give us our secondary sex characteristics. As well, we design anti-inflammatory drugs like dexamethasone here based on this skeletal structure to reduce inflammation from injury or to reduce inflammation from asthma or allergies. So understanding the differences between the different types of polycyclic systems helps us understand something more about our own biology and about drug design. On this slide, I've just shown examples of that. So here are 3 steroids that are biologically important to humans. Cholesterol on the upper left, estrogen on the bottom left, and testosterone on the bottom right. Over here, we have that 4-ring skeletal structure that's common to all steroids. If we just superimpose that over these 3, you can see that although there are lots of superficial differences between the 3 steroids, they all share that common 4-ring fused polycyclic system. It's the differences in the functional groups that each of these molecules have, as well as the substituents coming off them, that give them different biological and chemical reactivity. So with that, this pre-lecture is concluded. Be sure to review the relevant reading in the Carey textbook, and as always, make sure you come to class prepared to apply what you've learned.